I'm the session chair today. My name is Ian Lycap. I am from the University of Notre Dame, and I am the Research and Facilities Program Director of the Materials Characterization Facility there, which is a handful of lots of words. I wrote everything I'm going to say, which is just like a minute, just because when I try to improv stuff, I just always regret whatever I say. So today's session, we'll have a talk from a sponsor, and then we'll have 3 speakers from different universities who are doing some work in the life sciences areas, but they're doing that with techniques that are typically associated with more materials analysis. So the goal, hopefully, like you might be materials analyst yourself, but hopefully if you're in the bio field, the goal is to show some examples of things that you might be able to share with your researchers and maybe able to use in your own research as well. All right, so the very first thing I have to do is housekeeping. So I'll just read these. This is good for every session. So 1, please silence your phones. Photography and video recording of sessions are prohibited. Recordings of the program sessions will be available on the meeting website following the annual meeting. 3, ABRF's social media policy is that everything can be shared. If a speaker asks that it not be shared, please respect the speaker and do not post on any social media. The hashtag for the event is #ABRF2024. Unless noted by the speaker, questions are to be held until the end of the session, including questions for the author of the lightning talks. Attendees may ask questions at microphones in the room. We're going to do that differently here, so we'll have a little bit of time for questions at the end of each talk. There's a plug for the meeting app. I'm going to skip that, sorry. In that meeting app, though, sessions and lightning talks can receive a 1 to 5-star rating in the app. In addition, all attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. All right, so I think I will introduce. Well, actually, let me introduce our speakers after we do the talk from Tony. Tony's going to give a quick 10-minute talk, and then I'll come back up and introduce the speakers. So, go ahead.
Thank you. Great. Thanks for being here this morning, and thanks especially to Biomedical for the kind invitation to speak on their behalf and tell you about some of the work we've been doing with them at the McGill Genome Center on trying to maximize epigenomics and multiomics work at our facility. So I work at the McGill Genome Center, which is a genomics core facility up in Montreal, Canada, and one of my primary interests is in epigenetics and epigenomics, and where I manage the McGill Epigenomics Mapping Center. And this is part of a larger national and international initiative to basically generate full reference epigenomes and then provide this data to the wider research community. And what is epigenetics or epigenomics? So for us, it's number one, basically the genomic sequence of DNA, but also trying to interrogate the methylome, the transcriptome, which is gene expression, as well as chromatin accessibility. So through my involvement with the Canadian initiative, that's where I first met Biomedical, who had a product which was actually kind of quite interesting to us and could potentially solve some of the problems that we'd been seeing at the mapping center as well as the genome center in general. So their product is the Duet Multiomics Mod C solution, which basically generates the 5-base genome, which is the genomic sequence, the A, C, G, and T, plus the methylation. And what's interesting about this solution is that you can start, it's basically a very simple workflow where you just take a small amount of input DNA down to 10 nanograms, you make a single library prep, you sequence a single library, and then you get both the genomic sequence as well as methylation. And then they put it through the pipeline and you get your multi-omic analysis. And this replaces conventional workflows where you'd take even more DNA. You'd have to make 2 separate libraries, a whole genome library, and a bisulfite converted library, sequence both of these libraries, and then stitch the data back together again to get your epigenomic analysis. So as I said, this potentially solves one of the problems that we've been seeing at our mapping center and at our genome center in general, which is more and more people were coming to us with more challenging samples. For example, FFPE or cell-free DNA, or they come to us with tissues where it's just hard to extract a lot of DNA or RNA, or it's hard to go back and resample patients to get more material. And the researchers still want to try and extract as much information as possible. So this solution seemed really interesting to try and solve that issue. So normally when a company comes to the center and they have a product that they want to try, normally we do an internal bake-off with other products to see how it performs. But we decided instead to actually have a workshop where we basically invited participants to provide their own real samples. We basically put it through the MOD-C workflow, and this workshop was done in collaboration with Illumina, where they provided the sequencing in kind. And basically, we just give the data back to the participants. And what we saw was that the participants had quite a wide variety of different sample types, some from cell-free DNA, some from genomic DNA, different cell types and tissues, as you can see. And for the species, we got human, mouse, and we also had stickleback, which is a fish species. So the first thing we did was, okay, how does it actually compare to, just at the genomic DNA level? So first thing we did was we just looked at the variant calls. We took a reference sample, which is one of the genome in a bottle samples, the NA12878. We looked at the variant calls using standard WGS as well as the MOD-C method. We can see relatively similar numbers of SNPs called, and there's about a 94% concordance between the SNP calls between the 2 methods. And this is in line with what biomodal had been seeing internally. And this is only at 16x coverage. I suspect once you go higher, we're going to get better concordance. But of course, we're not interested just in the genomic sequence. We want to look at the methylation. So the first thing we did was to look at basically the CpG calls. So if you look at the first, the first 2 rows of this table, we took a human embryonic stem cell line where we did the classical WGBS method and the MOD-C. And as you can see, the MOD-C just outperforms the WGBS. Both were sequenced to around 16x coverage, but we're actually detecting more CpGs, and these CpGs are also covered at higher read depth. But what's really cool about this is that we use far lower amounts of input DNA, down to 65 nanograms for this. So it's about 1/20th of the amount that we use for standard WGBS. And we see similar performance using cell-free DNA from a renal carcinoma cell line. If you look at the methylation patterns, and this is just a screenshot from IGV, you can see that for both methods, the MOD-C also detects the same methylated and unmethylated CpGs and regions. Next, we look at the correlation of methylation calls. I'll just point to the right side, which is the CpG islands. And the CpG islands, I think, are more interesting than individual CpGs, since the majority of gene promoters reside within the CpG islands. So the correlation is actually really good. It's a Pearson correlation of 0.94 between the methylation calls of the 2 methods. So to summarize, for the MOD-C, it's the cost is actually comparable if you're trying to get the variant calls as well as methylation data, but this is where it stops, and The MODC is better in a lot of other aspects. The bioinformatics processing time is much quicker. You can go down to very low input amounts, and it also covers a higher number of CPGs, and these CPGs are covered at higher depth. I'd like to point out that Biomodal also did a separate, I guess, collaboration with Illumina with the MODC workflow, and this data is available on the Illumina base-based sequence hub. So as I mentioned, epigenetics is not just methylation. It also includes the transcriptome as well as chromatin accessibility. So we're really excited about this new kit that Biomodal just recently released, which now instead of the 5-letter, it basically interrogates the 6-letter genome. And this one, it's in addition to methylation, it can assess the hydroxymethylation. And what's interesting about hydroxymethylation is that regions containing 5-HMC are known to be turned on or poised, resulting in increased gene expression. So this 6-base genome solution is called the Duet Multiomics Evoke solution. And because of the 5-HMC, what we see is that for the Evoke, it does basically everything that the MODC does, but now with this 5-HMC, you can now also infer gene expression, infer chromatin accessibility, as well as active versus prime versus repressed enhancer states. So we're really excited about this. We also did a workshop with Biomodal with the Evoke kit recently. And the samples, I don't have the data yet, but the samples are actually in the queue at our center to be sequenced. So again, I'm really excited about seeing this data and seeing how it can capture all this information. And again, for the Evoke, basically it's from a single sample. You sequence one sample and you get all this information, which replaces, in this case, the conventional workflow, you'd have to make at least 5 libraries, sequence all 5 of those to be able to get that kind of information. And finally, I just want to talk a little bit about the McGill Genome Center. So it's primarily a genomics core facility, but recently, over the last few years, we started moving into proteomics as well as metabolomics. We have a full bioinformatics team, so we're aiming to be this sort of one-stop shop for everything, A to Z. You give us your sample, we'll basically process it, do the sequencing or whatever is necessary, and we can either give you the raw data or we have a full bioinformatics team that can actually do the analysis for you as well. And finally, I just want to thank people at Biomodal, Illumina, and Genome Center for helping with the workshop and this collaboration. Thank you.
We do have time for a few questions.
Okay. Any?
I know I have one as someone who
Sure.
doesn't do this every day.
Right.
And if I work with the person who does genomics at Notre Dame, on a different committee. So if I were to say the key takeaways here for me was it just seems like it's so much more efficient as far as time.
It is.
And I noticed it can do nanograms.
It can. So we went down to 10 nanograms for the cell-free DNA. I mean, it worked quite well. It's the sequencing cost it's roughly the same, but you just get more information as well. And I think it's also a higher quality information. I think it's really, to me, it's a game changer for people who they're coming with samples that just don't have very much input, everybody wants to have they come to us and they say, well, we have 50 nanograms of DNA, and they want to do all these different things. Like before, I had to tell them, I'm sorry, we just can't do that for you, right? But now I think, with this solution, we're able to do that, and we're able to give them even more information than they thought possible. So I think it's going to help a lot with those types of samples.
One more question.
I was just wondering, can you talk a little bit more about how you infer the chromatin topography using this kit as well? Because I noticed that on the last slide.
I guess the best answer for that would be to go speak to the people at the Biomodal booth. actually right there in the back row. They have way more technical information on that than I would. Okay.
All right, thank you.
Okay, thank you.
So our next speaker is Marisa Asadian. She's actually a graduate student at the University of Illinois Champaign. She's in the Sweetler lab right now, and her work is in the development of techniques and methodologies for studying biological systems using image-based chromatography technology. And today she's going to talk about some of her work in alterations in Alzheimer's, the Alzheimer's disease model through multiscale mass spec imaging, which I find to be incredible. I do some mass spec with gas chromatography, but the imaging is really cool. So I'm one of two very
Okay, hi everyone. As Ian mentioned, my name is Marisa Asadian, and I am a graduate student at University of Illinois Urbana-Champaign in Professor Jonathan Sweetler's lab. And today I'm going to talk to you guys about, we just had a talk about omics and spectrotranscriptomics and proteomics, so my talk is a little bit on lipidomics and basically using multi-scale mass spectrometry imaging to explore biochemical alteration in Alzheimer's disease and its usage in neurochemistry. So multiscale mass spectrometry imaging is basically matrix-assisted laser desorption ionization, which also stands for MALDI. And this is basically a surface imaging technique that utilizes organic molecules such as 2,5-dihydroxybenzoic acid to assist and help laser desorption and ionization of the analyte. So here I have this metal target plate, and I have a solid co-crystallized matrix with the analyte. So we apply a few nanosecond pulses to the co-crystallized matrix on the analyte, and what it does is that it causes desorption and a transition from the solid state into the gas state, and through an unknown transfer of energy mechanism, it causes ionization. We actually still quite don't know how this mechanism quite works, but there is a transfer of proton, which then causes ionization of your analyte, and then it goes through a mass analyzer and then a detector for contaction. So this is a soft ionization technique, and it's you can basically use it in studying biology and probing biomolecules because it doesn't cause any fragmentation, which is very useful in studying biology. Here, what I'm basically showing you guys is actually the metal plate with a slide adapter. So this is very useful in studying biochemical processes in biology. So all you do is you have your fresh frozen sample, a thin layer of the fresh frozen sample that is applied to the glass slide, and then you apply the matrix, and what it's very useful is that you can actually probe an intact tissue without further sample preparation. And not only that, you can get spatial distribution of the biomolecule analyte as pixel information. So this is where basically mass spectrometry is imaging technique. So this is our Bruker instrument. This is the one that I use. It has multi-functionality, but I only use the MALDI for my purpose, for my studies. It has 10 kilohertz of frequency, which provides 15 pixels per second data. And not only that, what I do like about this, and the reason why I use this is because of the fact that it has a tunable laser size. So you can adjust the laser size, and it goes down to 5 micron spatial resolution, which basically allows us to even look at single cells and look at tissue with high resolution. Okay, so in our lab, we utilize this mass spectrometry imaging, and not only that, we have developed a variety of different techniques to look at basically single cells, so single cell level imaging with a platform called micro-MS, which stands for microscopy guided mass spectrometry. And also there has been a lot of computational and deep learning development in our lab. I am not going to talk about this a lot, but it was just published in Nature Methods not too long ago. And this is basically a method where you can do 3D reconstruction reconstruction of the brain using mass spectrometry imaging and this deep learning technique, and you're welcome to check out the paper. Okay, so now that I have shown you guys different ways of doing mass spectrometry imaging, what I am studying, along with a few collaborators, is looking at alteration in lipids and lipid metabolism in Alzheimer's disease. So this complicated image that I have here is just to tell you that lipids are very important in the brain. They are one of the most abundant class of molecules in the brain by dry mass, and they have a lot of physiological significance in the brain, which makes it actually an area that we are very interested in probing and looking at. So we're basically using our MALDI technique to investigate pathogenesis of Alzheimer's disease and how the lipid metabolism changes with progression of Alzheimer's. And for those of you who have never seen a plaque or an Alzheimer's disease plaque, this is what it looks like. This is just like a histology image, and it's basically, these are just like fibers of proteins, and that is basically misfolded, and it's in the extracellular region of the brain. So we're actually going to take a look at them and look at the lipid metabolism within that. So a postdoc in our lab, Dr. Timothy Trankline, has developed this computational strategy that you can use what we already have, so we can actually stain the plaques. And we can do this using microscopy images. And he found a way, or he developed a way, to co-register the fluorescent image of the tissue with the mass spectrometry imaging. So what you're seeing here, these are actually lipids that are aggregated in the region where the plaques are formed. So now, in the combination of having the two combinations, we can look at the lipid distribution and how it's associated with Alzheimer's plaques. And here you can see like a large amount of aggregating where the plaques are. But in the bottom picture right here, what you see is actually a depletion of a specific, this is a sterile lipid. So you can see depletion of this lipid where the plaques are formed. So we can get this spatial distribution of this, of lipid metabolism and lipids in the plaque region using this strategy. Additionally, what we can do is, using this co-registration, we can look at molecular changes that occur in Alzheimer's disease. For example, in a wild-type brain, what you see are these phospholipids. However, in the Alzheimer's disease model, what happens is that you see a cleavage version of this phospholipid as lysophospholipid, and these are very abundant and one of the features that we have been able to identify in Alzheimer's disease using this co-registration model. Okay, so now I'm going from tissue level to single cell scale, and in order to do that, I am introducing you guys to a fluorescent microscopy guided single cell mass spectrometry. This is very high throughput, and this is how it works. So we do have a research professor in our lab who dissociates the single cells from distinct regions in the brain, and he plates them on this glass slide. This glass slide is then imaged using a fluorescent, just like a regular fluorescent microscopy. These slides also have fiducial marks, which then we use to convert the pixel coordinates from the fluorescent image into a physical coordinate of the adapter target plate to basically navigate the laser. So once we do the fiducia training, what you get is that you're basically telling the laser where to go. So I can tell it to go and probe that single cell, which I have basically this, I have a list of target, which is basically my single cell. So it goes there, and then it goes and probes the next single cell, and then it goes on and probes the single cell, another cell on the slide. So this method is not only a single cell method, but it's high throughput because, for example, in a glass slide of 25 times 75 can be imaged in 20 minutes, but if I were to do just a simple mass spectrometry imaging, that would take about 48 hours. I did this calculation based on 80 micron resolution, so you can imagine that if you were higher or lower, these numbers would change. Okay, so along with the single cell mass spectrometry, what we can do is we can also add immunohistochemistry in addition to this single cell probing. A colleague of mine, they have developed this antibody conjugated mass tags, I would call it peptide tags here too, which basically it's just an antibody conjugated to a peptide mass reporter, and it's introduced during doing just a simple immunohistochemistry, which then you apply a UV light, you cleave and release this peptide unit right before doing your mass spec analysis, which then gives you biomarker information and actually protein information of single cells doing mass spectrometry. What's so cool about this and what's so interesting about this is that you are not limited by your fluorescent channels, which is one of the major limitations in fluorescent microscopy, where you can only conjugate 4 to 5 fluorescent probes to antibody. Here you can do as many as you like, because as many as mass tags you can produce, you can have antibody and biomarker information. And here I'm just showing an 8-plex version of it, but you can go up to a higher resolution. And what are you looking at? I like to not assume that people know what they're looking at. So each channel here represents an ion distribution of each of the peptide tag acting as a mass reporter for a biomarker of interest. This is basically, it's a very low objective or low resolution image, which doesn't really matter for our purposes, but these are basically single cells, and you can see distinct spatial distribution of these mass reporters. Additionally, you can utilize this for Alzheimer's studies. So I do have, I'm showing here 3 different biomarkers, and you can see their spatial regulation in, spatial distribution in wild type mouse and in the Alzheimer's mouse, and you can see how these are upregulated in the mouse model. So this becomes very useful in studying Alzheimer's disease. And what we can do is we can also dissociate single cells from the tissue of a specific region and also do and study Alzheimer's disease with mass spectrometry imaging at single cell scale. This is basically a spectrum of a cortex region of the brain of an Alzheimer's disease compared to its wild type. Okay, so now that we have both biomarker cell classification ability and the fact that we can look at lipids, what I have developed is a 2-step approach where you can actually first just do a lipid profile or lipid targeting of the single cells. Here I'm showing an average spectrum of 1,300 single cells, and these are, you can see that we can identify a variety of lipid features. These are just a few of them that are highlighted, which are some of the phospholipids that are abundant in biological system and they're also dysregulated in Alzheimer's disease. So, additionally, we can add our immunohistochemistry into this method by targeting the same single cells that we targeted for lipid profiling again, and basically get its biomarker distribution. So this becomes very, very useful for us to be able to do cell classification based on 2 information, lipid and also biomarkers and proteins. And the purpose of this project, which I'm still quite working on and I'm trying to finish up, is to do that, to do cell classification based on biochemicals, specifically of a single cell, so lipids and biomarkers. And as I'm showing here, you can see that I'm showing an astrocyte, which is like a particular single cell in the brain, and we can use them. We can use specific biomarkers that are specific to the specific cell. You can also have neurons. They have a variety of different biomarkers, which then allows us to subclassify neurons in the brain. And I'm also planning to implement this in Alzheimer's disease. And so yeah, so as right now, all I'm doing is basically trying to do single-cell classification just in the neurons in the brain single cells, and also apply that to the Alzheimer's disease model, which I have shown here. Additionally, just to give you guys a summary, I have also shown you guys experimental and computational framework for enhancing mass spectrometry imaging data by probing both Alzheimer's disease brain tissue and compare it with, in comparison with wild type. And the whole idea is to take the tissue information and the single cell information to basically provide a comprehensive biochemical changes that are occurring in the brain, in the Alzheimer's brain, and compared to the wild type, and with single cell specificity. So this is all I have. I wanted to thank you guys for having me here today and for this opportunity. Here, this is my professor, Jonathan Sweedler, and also our collaborator, Professor Fan Lam, who also helps a lot with the computational modeling. And yeah, with that being said, I will be happy to take any questions you may have. Hi.
Yeah, thanks. So I don't come from a mass spec or MALDI background, but I was curious if you could talk a little more about the sample preparation of having a full slice of tissue from what I took on a slide, when what I know about it is mixing the matrix and analyte and spotting, and how do you make sure that you're ionizing everything you want versus something that just preferentially desorbs when you have such a complex sample system?
So yeah, it's really hard to do that. So you are going to get a spectra of not just your analyte, but also your analyte and the matrix itself. And you also get different things, but you haven't, there's like a database as far as lipid goes that you can confirm what lipids you're seeing. And not only that, you can also do LC-MS so that you can confirm that you are seeing a specific lipid by using standard calibrations. So you don't get a mass spectrum. So here, if I show you half of this, let me see, half of this, it comes from the matrix. This is one of our biggest problems, is the matrix can cause low signal to noise ratio. You do see a lot of its peaks, but with the mass tags, I go in knowing what to expect and what I'm looking for, so I know which peaks to look for. But with the lipids, you do have to do further analysis. You can do, I guess, MSMS, like fragmentation. You can run standards and calibration. And there's also a lipid map, which is basically a huge computational and LC-MS database that you can go from. So, but as far as sample prep, I feel like I didn't understand. The sample prep is just what you do is, I don't know if I can go back. I have so many animations. So in the sample prep procedure, just this cool crystallization, you can do sublimation, recrystallization, where you mix like organic solvent, for example, with your matrix, and then you spray it onto your sample. They're just like, they're specific instruments that you can use, and then what happens is the solvent evaporates, and then a crystal lattice is formed, which is basically the cool crystal of the analyte and the matrix. Yeah. Hi.
Great talk. I had a question about the mass tags. Are those something you're designing and synthesizing, or are they commercially available?
So yeah, they are commercially available, and so I put the paper here, but it's Ambergen. It's the company, and they're one of my collaborators. So they basically give me the mass tags that I need, and I do the rest, basically, yes.
How many plex can they go up to?
I think, oh my gosh, I think they even up to 70 maybe. You can do more. As long as you can see them in your mass M/Z range, there's no limitation. So, yeah. Yeah. Thank you, guys.
All right, thank you again.
I don't know how to exit. I can just close it. Okay. Okay.
Yep.
Would you,
Okay.
oh,
Yeah, you can start. That was a great talk. I feel like I had a question on like every slide. As someone who doesn't ask that, I understand what some of the problems are. But I get why you're making these mass spec probably just to see like, oh, that's that.
yeah.
Our next speaker is Abigail Lytton-Jean. She's from the MIT Koch Institute for Integrative Cancer Research. And her facility addresses nano-characterization of biorelevant materials with an emphasis on multiscale cryo electron microscopy. And did you get the computer? Okay.
Ah,
All right, have at it.
shocking, right? Hello, everyone. Thank you for being here. Thank you for inviting me. And that was a great talk to follow, by the way. Thank you. That was really nice to see. I have a lot of ideas to maybe collaborate with you, actually. So just sort of a small introduction to where I am approaching the work that I do. I'm a chemist by training. I did my PhD just slightly north of Marisa up in Evanston at Northwestern University with Chad Merkin, where we were working on gold particles that were decorated with, thank you, different biological molecules, and using these materials for programmable self-assembly, using these materials for diagnostics and detection systems, drug delivery platforms, and then understanding the new material properties that were emerging from the work that we were doing. I then transitioned to MIT for my postdoctoral work, where I worked with Bob Langer and Dan Anderson in a slightly different kind of lab, a tissue engineering lab, where we were focusing on synthetic nanoparticles for intracellular drug delivery. We all have our nice Moderna vaccine, I imagine. Other types of work looking at how the tissue engineering world could devise applied, I guess, materials more in the direction of nanotechnology. Anyway, so as I was wrapping up my postdoctoral work, I had the opportunity to build a new kind of core facility from the ground up. And I thought this was a really exciting opportunity because I got to build what I wished I had always had when I was doing the kind of research that I was involved in. So we think of the research that we are supporting as the nanotechnology materials research that happens at the interface of biology. And so the lab that has formed very organically around the needs of this community. And we are often asking biophysical type questions like, how big is something? What shape is something? How many have we made? How soft or squishy are these materials? Where are these materials located in the larger context of the research that they're in? So I thought I would take us through a little bit of a tour of how researchers may transition from one kind of capability to another kind of capability within our core facility, because the selection of instruments and capabilities that we have is sometimes a little unusual combination, and it might not always make sense at the onset, but we have people doing work where they happily rotate through all of these different kinds of capabilities. So frequently we have a lot of people doing nanoparticle work. So a very first pass, quick, easy way to approach nanoparticles when people are trying to understand what they've generated, what size particles, is to introduce people to DLS measurements. A quick, easy way to just screen large libraries that people may generate to get a sense of, do we have small molecule aggregation? Do we have large aggregation? Do we have specific populations of particles forming? Once people screen their samples, they can then whittle it away down to your hits of interest, of the samples that you're most interested in. And I will introduce people to a nanoparticle sizing and concentration counting instrument, Spectrodyne. This is a new technology. We were, I think, the 4th instrument they had ever designed, and you can take a sample that's a complex mixture of different particles. You may get a DLS curve that looks like this green line, but then when we introduce these samples to this kind of instrument, we can have a far better resolution in terms of the different sizes of populations of particles, and critically, we can count concentration in terms of particles per mil. It's a very simple little question to ask what the concentration is, but in the biological world, it's really, really difficult sometimes to get at that information. We will also have people who want to understand, what shape of nanoparticle have I made? And so we will introduce people to our AFM. This is an example of some DNA origami work where some of our community was designing these DNA assembled structures to fabricate much larger, complicated structures. Some of our people, after generating their nanoparticles for therapeutic drug release, they will then transition into working with hydrogels, and they will often introduce these nanomaterials to hydrogels, or maybe they're just working with hydrogels that don't have any nanoparticles present, but they're generating these new materials for tissue engineering, where they want to use a scaffold to grow cells. And so they ask, how stiff is my material? How squishy is my material? As the nanoparticles are releasing drugs, how is this changing some of those, the modulus of a sample? And so people will use the rheometer to just start looking at these samples in sort of a bulk measurement. So these are instruments that I would say they're relatively accessible. I can train your community in either just a single session or several sessions over the course of a week, and then people can be relatively independent and they can work on their own terms. The rest of our lab is heavily focused on electron microscopy imaging. And for anyone in the biological electron microscopy space, immediately that most of the work is done in the sample prep side of things. And so for all of the instruments that you see here, we have a low kV TEM, we have a higher kV TEM that operates in cryo mode, we have an SEM fib that's in room temp and cryo mode, and we have a confocal microscope, also room temp and cryo mode. For these 4 instruments that you see here, we have over a dozen sample preparation and sample transfer instruments that are all necessary to get samples to this endpoint imaging stage. And so while these are the most flashy instruments, the most expensive instruments, they need their own rooms, I would say that a lot of the magic that's happening is behind the scenes in terms of figuring out what type of workflow, how are we going to process this sample? And frequently people are going to work with many different workflows to determine different aspects of their samples as opposed to just sticking with one. And so this is a complicated slide. Don't spend too much time looking at it. It's not even complete. I just mention it because this is where I feel like we put a lot of our energy. So before I get into what some of this data can look like, I thought I would just make a brief comment about biological cryo-EM and the different length scales that this can happen on. Some people, when we say the word cryo-EM, they immediately think of structural biology, single particle protein characterization in this area. And this is a very important part of cryo-EM, absolutely, but it's only one small sliver of a large length scale where cryo-EM can happen. This can include looking at nanoparticles and engineered drug delivery. This can look at subcellular imaging or perhaps cellular imaging in the context of the materials that they are growing in. If anyone attended Kirk Zimick's talk earlier, expanding into the volume EM space where we're now looking at 3D cellular tissue reconstruction, even all the way up into the connectomics world where you're imaging an entire Drosophila brain, just amazing expanses of data that are generated. And so we really focus on this area, but we also generate some sample processing and prep that feeds into more of the structural biology. So At this point, I didn't want to talk about the little details and nitty-gritty of workflows and instruments. I just sort of wanted to give a snapshot of the kinds of flavors of imaging that we can generate. So we have a lot of people that work with nanoparticles, and of course, frequently just doing room temperature negative stain TEM is a great place to start. But then very quickly, people will transition from looking at these materials at a room temperature TEM and looking at them in cryo TEM. And you can very quickly see that the structure of the morphology of your nanoparticles is quite different when you're looking at a sample that is preserved and hydrated. And these are just, more beautiful images of what different types of biological sample particles can look like in cryo mode. Nanoparticles are convenient to work with in the EM space, TEM space, because they're small, but we also, actually, I forgot to mention, this is actually key for this talk, is that in a lot of our nanoparticle work, we're also doing more materials type characterization. And so this is just sort of one snapshot of saying, well, not only do we care about the morphology and the structure of our particles, we also want to understand what is the elemental composition, and so we provide a number of different analytical capabilities as well. Moving on to samples that are larger and take more work, there are numerous types of resin processing techniques where people will work with cells, tissue, these larger materials to embed them in resin. Now they're in a hard state where they can be sectioned into thin sections, amenable for TEM imaging. And so this is an image of liver tissue where this is obviously false colored, where each different color represents a different cell. Doesn't start out like that. This is more of the kinds of imaging that you would expect of tissue samples, this being brain tissue. And for some research, this is the answer. This is what they need to answer the questions they're asking. This is a 2D image. It's like taking a single slice out of an entire loaf of bread. But for some of our community, they actually want to image what does that entire loaf of bread look like. And so as Kirk had mentioned earlier today in the initial sessions, we can then work with these samples instead of in the TEM, we can work with them in the SEM fib. And so using the fib, you can mill off a few layers of material, exposing new sample underneath, where we then do more imaging and iteratively mill an image. And so when you then assemble this stack of thousands of images, we can now look at cells in a three-dimensional context where we're looking at the nucleus, and we see different types of organelles and mitochondria. And you can do segmentation and processing so that we can now start understanding in a more three-dimensional context how the cells might interact with each other in addition to just the subcellular components. So this is a more advanced use of the SEM fib. We also do a lot of more traditional SEM imaging with, there's nothing biological about either of these samples. This is a microparticle that was engineered for nutrient encapsulation and more materials samples that were still relevant in a lot of the biological research that our community is working on. But we usually very quickly come back to biology. And so we will also do work looking at cells that are actually at room temperature. They're dried on a surface. Again, a false colored image of a cell, but this may be more of what it looks like to begin with, where we can see a collection of cells and then we can look at a particular cell at a higher resolution of the surface detail. And frequently people are always saying, okay, what's the smaller features? What are the tiny structures on the surface that we can look at? But we also have people that are sort of going the other direction that want to see larger fields of view. And this is really exciting about the SEM because you can look at really tiny features down into a couple nanometer scale, but we can also look at huge fields of view. And so people might say, okay, that's nice, this is my single cell, but I'd actually like to look at a family of cells. And you can quickly see that this project, they were generating small cells and they had larger cells. Maybe you want to look at a extended family of cells, or you could ask to do even better statistics and look at hundreds of cells. And if you really want to do strong statistics, you can look at thousands of cells. And it's important to appreciate that if you want to, you can generate these images where you're looking at huge fields of view and also maintain really incredible detailed structural information if this is important for the work that you're doing. That said, of course, we have people that want to do the small stuff. And so this is an example of malaria sporozytes that we had one researcher was working on, and she was interested in identifying the presence of particular antibodies that were present. And so each little white speck that you see here is a gold nanoparticle that is binding to the malaria sporozyte due to an antibody interaction. We can also do workflows where we utilize fluorescent imaging. So CLEM is a terminology that you will hear thrown around sometime. CLEM just stands for correlative light electron microscopy. It's a very broad umbrella terminology, can account for many different kinds of workflows, anything where you're doing upstream light microscopy imaging to target regions of interest for downstream EM. And so in this case, they were looking at which sporocytes had interacted with different fluorescent antibodies. And so, again, this is a sample that's dry, it's at room temperature, but the next step was to then say, okay, what does this really look like when it's still hydrated in its native form? And so we then worked with her to do a cryo-CLEM workflow, in this case, cryofluorescence to cryo-SEM to cryo-TEM, where we're looking at an entire TEM grid here. We've mapped it out. We can zoom in on just this region, and then we can zoom in a little bit more to just look at this single grid square, where here we have 2 malaria sporozytes visible via the fluorescence. You can imagine this would be really difficult to find those sporozytes if we didn't have fluorescence present. And then she said, okay, well, I really care about the very tippy tip of what's going on in these sporozytes. And so this was transferred to a cryo-TEM for imaging at the same spot. And this was done in collaboration with Gang He, who's with Wachu at Stanford. So these, like nanoparticles, they're convenient and easy to work with in the TEM world because they're small. We didn't have to do anything to this sample in order to make it thin enough to image in the TEM. But that's not the case for everything. What if we're working with yeast? What if we're working with cells, tissue? And so we will apply other workflows. It's still similar to the cryo-CLEM workflow as well. We're going to see the similar concept where we first are mapping out a frozen grid in fluorescence. We find our one region of interest, and then we transfer this into our cryo-SEM fib. We overlay this fluorescent image with fiducials in our SEM to say, okay, that is the region we care about. And then using the fib beam, so the fib where we generate, a few slides back, there was that movie where we had milled an image to create 3D understanding of entire cells. Now we can use that same fib beam and we can mill out sample above our region of interest, do volume imaging in the process, and then mill out material below, leaving just a thin little lamella that's only 200 nanometers in thickness. And now this is thin enough to image in the cryo-TEM, and it should be of the region of interest that we were interested in the first place. So this was a really fun collaboration with Kirk Zimick, who was at Zeiss at the time, who is now running his own lab at Danforth in St. Louis, and also with Gang He and Patrick and Wachu. This looks blurry. Anyway, so they're at Stanford. And so the data ended up starting something like this. We were looking at the fluorescent regions of interest on a cryo sample. This sample would move into the SEM fib to mill a surface. And then you can overlay that fluorescent image to find your regions of interest. If you take this sample and you tilt it 90 degrees, so you're looking at it from the perspective of the fib beam, it looks like this. You've trenched out the fib above, everything below, and now you have this thin lamella. And on FOSS, you can see everything that's contained in that lamella. And then this is transferred to a cryo-TEM for cryotomography. And this is where you can then begin moving in the direction of asking those structural biology questions. What's happening at that very fine angstrom scale? So this is my team of really fantastic scientists, and I would take just like the last 60 seconds to make a comment about staffing. And I know that different types of facilities operate with different models. For the kind of work that we're doing on this EM side, this is really difficult work. It takes really experienced people who are creative, they're innovative, they're constantly developing new workflows, working on protocols. Every person who walks through the door to work with us on an assisted level, they are getting a bespoke, tailored approach to just their project. And to attract people like this, and then to retain people like this, it means having appropriate job descriptions. It means having a career path so that they know that their hard work is appreciated and they're being rewarded for it. It means having a good performance evaluation system so that they can have appropriate salaries. And I keep stressing to point out how different the people in my lab are from people who are in academic research labs. Because most of the people in an academic research lab are probably a graduate student, a postdoc, a technician who is getting ready to go as a stepping stone to med school. They have short-term appointments. And so the people in my lab are so different because I hope, fingers crossed, that they are going to be with me for a very long time. And so this means having a career path that rewards them and recognizes that while on paper their job responsibilities may not change, they are growing and expanding with every project they encounter. So I would leave it at that and say thank you for your attention.
Okay, so time for just maybe one question. If you have more questions, you can always find these speakers after too. But so, does anyone have a question?
Come to the microphone. Yeah, thanks so much for that talk, and I couldn't agree more with the last point you made. I had a question about your AFM, which you kind of touched on very briefly. So you use it for kind of molecular imaging. Do you use it for viscoelastic measurements as well?
Yeah.
Biomaterials?
So AFM is such a powerful tool. I mean, wildly powerful. You can do, I mean, what I showed, that was just a very basic, like, here's the topography of the surface we're looking at. But yes, you can use it for looking at point force adhesion measurements, how tightly something binds to the surface, how stiff or squishy your surface is. What I find in our institute is that most of my community, while they are interested in using the AFM for these kinds of much more advanced experiments, I would say, it's not the focus of their work, and they just want it for one little image. And then when they understand the work that it takes to get there, they go, oh, well, okay, never mind, I don't want to do all of that. So I think for our kind of research community, for the AFM to really flourish and do the best work that it can do, it would need to have somebody who is really dedicated to providing services around that. In the same way that we do around the EM side, it would be good to have that on the AFM side, but we just don't have enough people coming often to be able to put resources towards that. But. Oh, yeah, you should come on up, actually.
So you can actually impose either a vertical or lateral sinusoidal wheel with the probe and do that at various rates to get a viscoelastic response. I actually started on that as a postdoc 30 plus years ago, but we didn't actually have that capability directly. But what we've discovered was that we could actually change the scan velocity by 7 orders of magnitude and measure the friction force. And it turns out that it's very much like measuring tan delta over a range of, and then later we got temperature control and so on. So yeah, there's a lot of pretty cool viscoelastic things you can do.
We should absolutely
It was the perfect segue to the last talk, actually, so thank you.
Yeah.
Let's thank Abigail once again for really fascinating. Our next speaker is Haugstad from the University of Minnesota. He actually graduated from there with his PhD and then his postdoc there. And while he was doing his postdoc, he actually set up an accelerator for helium to do Rutherford backscattering, and has been in the characterization facility since then. And the other thing that he specializes in is in bio-AFM and AFM, and so that's what the last talk is going to be about.
Yeah, this is not coming up. It was just plug and play before to get it to work. Yeah. Yeah, just had to switch to HDMI to this. Let me try it. This. Choose. It can be either side principle. Ah, there it is. Strange. Thank you. Except it's showing up on the wrong. Let's see if I do it this way. There, good. So thanks for the opportunity to speak here to everybody today. I'm Greg Haugstad. I work at the characterization facility at the University of Minnesota. I've actually been there a long time. I'm not going to go through that, but by training I'm a condensed matter physicist, but in my postdoc I got involved much more in chemistry and bio things and wet things and so on. And I've been there ever since doing primarily AFM. We have various people that sponsor us at our site. I'll just show you a glimpse. We have various things within our institution that support us. We have 3 centers that support us as well, and then we have an industrial consortium that provides a lot of funding for sort of methods development work. So anyway, I threw the slide in during your talk because I thought I should probably give just a little bit of an overview of what we have. I'm only going to talk about AFM today, but we do have a range of groups, including quite a bit of cryo-EM. We do not yet have a cryo-FIP. We're getting one pretty soon. These are some of the newer systems, but we range from X-ray scattering for purely structural analysis through various EB methods, primarily SEM, TEM, and microprobe. A variety of proximal probes, which includes 6 AFMs, a scanning tunnel microscope, nano-indenters, etc. Some other spectroscopic methods, surface analytical, like X-ray photoelectron spectroscopy, etc. And then I also run this accelerator for Rutherford backscattering and other techniques. Anyway, so here's my outline. I'm basically going to try to give you a few snippets of things that we've worked on in the bio or biomedical realm. Firstly, just basically some what I think is fairly interesting topographic things, some things you can do with abrasion by the tip. And then I'll switch to an intermittent modality where you basically are approaching and retracting the tip from each point on the surface and getting mechanical rigidity and so on from that. Mostly just in air, and then seeing how much time I have left, I'll transition to some of the same kind of work done fully immersed in aqueous environment and more sort of soft core biological kinds of studies. So firstly, I thought I'd throw this up. So this is just my obligatory slide that I show all the time, just to explain how AFM works in the most basic mode of operation, which is contact mode, where the tip is always touching, so you're rastering the tip across the surface. It's in contact, and as the tip is riding over the hills and valleys, that would cause the cantilever to bend up and down, which would cause this reflected laser spot to move on this sort of 2 knife edge photodiode. So you have a horizontal knife edge and a vertical knife edge, and the horizontal one, a top minus bottom read, is compared against a set point that the operator chooses. So you're sort of choosing how much your cantilever is going to be bent up or down, and thus whether the force is large or small. And then whatever deviation or error there is from that, that goes into some electronics, which then changes the Z bias to the Z scanner. And it's actually the variable Z displacement through that sort of feedback mechanism, the variable Z displacement that is recorded as a number at each pixel. So AFM images are not images per se, rather they're matrices of numbers, where each number at each pixel is a physical measurement. Here it's a measurement of how much the Z scanner is displaced up or down to keep this constant. And this just happens to be a partially de-wetted film of polyvinyl alcohol on mica. These are a few nanometer high steps or ledges between the different areas, and This is actually the corresponding error signal, and I like to render it in gray to make people think that it's SEM. But basically, this is Z versus X and Y. This is DZ dx versus X and Y, because the greater the slope as the tip is moving, in this case left to right, as it bumps up over a ledge or down over a ledge, the more the cantilever is going to sort of deviate or bump above or below the set point, and you get sort of this illumination. It looks sort of like it's illuminated surface. Anyway, so you can go on. So now you have these matrices. Again, you're measuring topography. Z is a function of X and Y, and you can go ahead and 3D render that. This is actually a true aspect ratio. So vertical scale is the same as the horizontal. And this is actually of human hair. We did a bunch of work on lubrication with hair conditioners and that sort of thing in collaboration with Unilever a while back. You could also just go to a straight sort of color, and I should have said that in the last slide. The idea here is you've got a color gradient for Z that extends from sort of dark to bright through some range of colors, and that's sort of how you're rendering Z, is just as a color gradient. Here, my color gradient, if you look right here at the lowest spot, this kind of trench up to the highest, it goes from dark green to light green to sort of magenta or violet up to white. And this is about 100 nanometer change of height, but again, there's sort of a 3D rendering along with the color that's going on here. These terraces here are actually 3.2 angstroms apart, so you have exquisite Z resolution down to even sub-angstrom, whereas you still have a range this way, and you can go with, in this case, is even more, of as much as a few microns of height. The greatest that we can go with one of our AFMs is about 13 microns of height. But the lateral range is bigger than that. My third example here, firstly in air, is some work we did through our consortium with this company. The biggest site is actually in the Minneapolis area. And so through that consortium, we were working on some of their PVP coatings for catheters and those kinds of things. And this coating is supposed to make them lubricious. So when it's fully hydrated, it should be a lubricious gel. And in fact, this is what you can actually do this underwater or under buffer. So you can bring into a liquid cell. So now your sample, your tip, your cantilever is all immersed in the liquid. And what happens is the water penetrates this coating, swells it into actually a gel phase. So now instead of, megapascal kind of modulus up here or higher, you have kilopascal modulus down here. And it turns out you can actually, I'm not going to show it here, but you can mechanically map that. I did throw in one more slide. The image I just showed you is the dry case is here, and the wet case is there. But what I'm showing you now are a variety of these coatings with different degrees of UV curing. And it turns out this one works about the best in terms of being lubricious and not shedding particles into the body. But they didn't really understand anything about mechanistics. And we discovered that their industrial process in the factory produces a lot of defects in these coatings, which then in turn give all these sort of pockets of stiff and soft. So this is way more heterogeneous than they wanted. What's also cool is if you go to the uncross-linked case, the non-cured case, and you slide the tip over and back, you can actually pluck the individual polymer strands. You can sort of stretch, snap as you go back and forth. Whereas when you look at the friction data up here, it's actually, it doesn't have these kinds of stretches and snaps. And by the way you get the friction, oops, I'm clicking the wrong button. The way you get the friction actually, if I go back to that schematic slide, is the left to right. So now the vertical knife edge, if you're scanning back and forth like this, you have twisting on the cantilever, which moves the spot slightly left or right, and that gives you the ability to gauge and actually image the friction force. I'm not actually going to show you any so-called FFM, friction force microscopy, but this is actually how you do it. Is you get this every single scan line, and you get a friction image out of that. So that's kind of cool. And then lastly, in contact mode, you can actually choose to go to a higher force. You can push harder, raster a smaller region, and sort of dredge it up, kind of scrape it up. And here we're trying to measure the cohesive strength of extracellular polymeric substances in a bacterial biofilm. This is actually for wastewater treatment applications, but it could be any other kind of biofilm as well. You can see the actual bacterial cells, and then what we discovered is that the high force actually tears the EPS. And if you do this a bunch of times, you can gradually dredge out a deeper trench. You can calculate the depth of that, the volume excavated. You can calculate how much frictional energy that you've dumped into this to cause that much disruption, and thereby actually measure a cohesive energy density that's sort of in the nanojoules per cubic micron machine. So that's some of the things that you could do in contact mode with a combination of topographic and frictional imaging. Switching to the more intermittent contact modality, where we're doing this more for conventional nanomechanics. So what you do is you can always stop at a point on the surface and just do a simple force distance curve where in time you're rastering the Z-scanner, or you're ramping it up and down to approach and retract the tip from the surface. And then you're actually measuring the force signal by the bending of the cantilever. You would usually specify kind of an ultimate force up here, tell it not to go any farther than that. But anyway, so this is just an air jump to contact, push up, pull back, stick, snap away. So you have this adhesion that goes on. But if you want to actually image this, if you want to produce nanomechanical images with high pixel resolution, you want to speed this process way up. So instead of doing this sort of once per second, you would like to do it more like 1,000 times per second, so that you basically once per pixel in a sort of ordinary high pixel density image. So approach, retract. We have a high-speed data acquisition that captures the cantilever dynamics, and we can actually grab the lowest point here to get an adhesion image, so tip sample adhesion that is, or we can analyze a little bit of the part in the contact regime here to get at rigidity and thereby modulus. So anyway, here's this one quick example. This is actually done in air, but it was at high humidity to keep the bacteria moist. I actually don't remember whether these are live or dead bacteria, but anyway, we wanted to keep them moist to see nanomechanical differences. So it turns out this population of bacteria has a subpopulation that's very soft, and the rest of them are more rigid. My coworkers on this were perplexed by that, but also there's a difference in stickiness. So the ones that are softer are actually less sticky as well. So anyway, just one example of how perceptive this technique is and the fact that you can actually image with high resolution. You could also take the topographic data, right? This is Z versus X and Y. Render this in 3D perspective and then paint this data on top of that to sort of combine two forms of information. So you're seeing the 3D, and this is the true aspect ratio again, the 3D sort of topography here just from perspective, and then the colors are showing you what's softer and what's stiffer. And so lastly then, of this particular sort of in-air nanomechanical imaging, we did quite a lot of studies. This is one that was actually published, where we're studying polymer drug coatings. And we're actually doing this both in air, which is the top height image here, and underwater or under buffer, and watching how the drug diffuses out. So from this, we sort of determined that the drug was diffusing out of these circular domains, producing pockets with time. But it turned out that just looking at the original dry images, imaging the tip sample adhesion showed a very, very strong adhesion contrast. So the drug is much less sticky than is the polymer. The polymer is this complicated arborescent thing. And we've looked at these as more conventional linear polymers of the same type, and they don't have this very fine substructure that you see here. So you're really rendering sort of something on the order of 10 nanometer substructure that deals with the branch morphology, or that derives from the branch morphology of the polymer itself. Okay, so that's conventional nanomechanical imaging. And again, at each pixel, you're approaching and retracting, roughly once per pixel, and you're measuring, the bending of the cantilever up, the bending of it down, the grabbing of the tip here, so that it pops away, and then in real time, you're actually seeing the ring down of the diving board. You jump off the pool, diving the board into the pool, and you see the ring down. That's what you're seeing there. Sometimes there's interesting things. If you're pulling out of something that's really gooey, there could be some structure here through the first cycle of this that can tell you something about viscosity and other things. Finally then, how much time do I have? Racing through? Oh, okay. Okay, so lastly then, similarly, I just want to show you 3 quick glimpses from these 3 studies, 2 of which are published under fully aqueous immersion. So firstly, lipid bilayers. My collaborators actually did neutron scattering, and what they're doing is they're basically incubating synthetic polymers into lipid bilayers and wondering where does the polymer actually go. Well, the neutron scattering suggested that the polymer is actually penetrating somewhat into the bilayer, which was they were a little surprised by that. So then we did AFM to sort of corroborate what they learned from neutron scattering. And so what we did is we're actually pushing against these bilayers with the AFM tip. And in fact, what we did is we pushed, firstly we imaged them in another mode of operation, which is very delicate, called AC. I won't get into that. And what that showed mainly was that the phased images were sort of blank. So this gave us kind of this null result that the polymer was apparently very homogeneously interpenetrating into the lipid bilayer. But then we did the nanomechanics, and we went to high enough force that we could actually pop through the bilayer. So we're measuring the rupture, basically how much force was needed to rupture these things. And what we discovered was that breakthrough force actually was the same. There's a distribution of values, so we did this in a mapping mode. But the distribution of values was pretty much the same, whether it was just plain lipid or a lipid after it had been sort of incubated with these other polymers. So that corroborated the neutron scattering bit as well. And actually earlier in the project, we discovered that we were not preparing our samples properly. They were actually seeing air for just a fraction of a second before they were re-immersed in liquid. And that actually gave us quite a heterogeneous surface with a broad range of rupture forces. But I threw this in here because this is my colleague, Dr. Guishuan Yu, who writes custom MATLAB software for a lot of our methods to enable us to do special kinds of analytical things. And this is his software that basically produced these results. Okay, so that's the model then for what we actually have. Lastly then, sorry, a little long here, but we did this, again, very fast underwater to actually measure the stiffness of liposomes as a function of their radius. Again, we had to use MATLAB to do some very custom post-processing of data, but we did actually get this 1/, stiffness versus relative diameter, and so we can see that how these things do actually get softer, so 1/, stiffness goes up as you go to larger and larger diameters. So you can actually measure these things, even on small objects like liposomes. Very last, I'm not going to read this stuff, but I had to put this back up here to remind me what was going on in this study. This was published very recently. But we were studying Drosophila ovaries and measuring basically modulus. My biological collaborators refused to call it modulus. They wanted to call it stiffness, so this is from the paper. But it is actually modulus, so we're doing contact mechanics modeling, Hertzian contact mechanics modeling. So basically a three-sided pyramid pushing into these things. And the upshot is two populations of these ovaries, one which were basically defective because they're missing some critical biological or biomolecular component, the other which are not missing it. And these are the sort of the differences of moduli that we measured with the error bars that showed that indeed, you had to have this extra biochemical component in order to get proper integrity of these of the membranes of these ovaries. Okay, and that's just a little more of the same thing. I actually gave part of this in an AFM audience talk, and so I talked more about issues with our data processing. But anyway, you get a sense of the size of the Young's moduli that we're measuring here. Way down in the, I guess, tens of kilopascal. Okay, then lastly, I'll just throw this up here, and I'll just let you read through the blue text up here is kind of the things that I just briefly touched on. I showed you lots of little clips from lots of different studies, so there's a whole range of people that have been involved in this. I won't try to read any of that off, but I'll thank you for your attention.
Basically, it's a rudimentary AFM kind of measurements for the kind of people in my community who really don't want to take the trouble to learn the sophistication of AFM. It's basically an AFM with like a bead on the end of it, so you could look at the squishiness of like a monolayer of cells or a hydrogel that maybe is being used for tissue engineering, cell growth. And from somebody who's such a severe AFM expert, I was just curious if you've seen this or had comments.
I have not heard of that. So it sounds like it's very customized to do just this, to work, AFM type cantilever-based measurement, but with a microsphere at the end, which is much better for gels and in some cases tissue as well. So I'm guessing it's just much more automated in terms of loading samples and I have the same problem. We have lots of people that want to get these kinds of results, but hardly anybody wants to get trained to do it themselves. And I think that's part of what's behind companies like you mentioned, is that to try to produce something that the average person who doesn't want to spend months getting trained and become an expert on a conventional AFM can do with a more narrow targeted measurement. That's interesting. So that's just a company that just came out in the last couple of years or something?
Yeah.
Okay.
Yeah.
Interesting.
All right, well, thank you, and thank you guys for coming, and hopefully you enjoyed the 3 fantastic talks about, I think, really powerful techniques. So, one more round of applause for you guys.
Thank you.